Tiny cells, big impact. The world of monoclonal cell lines. These tiny cells have a monumental impact on science and medicine. Monoclonal cell lines are like tiny factories working tirelessly to produce essential substances. They produce important substances for research and medicine, including antibodies, hormones, and enzymes. These cell lines are derived from a single parent cell, ensuring uniformity and consistency. This means all the cells in a monoclonal cell line are identical, sharing the same characteristics and behaviors. They have the same genetic makeup, which is crucial for reproducible and reliable experiments. This makes them incredibly valuable for studying diseases, developing new drugs, and producing therapeutic proteins. Researchers can test new treatments on these cells to see how they respond. Monoclonal cell lines are essential tools in modern biology, providing a stable and consistent model for scientific inquiry. They allow researchers to study specific cell types in a controlled environment, eliminating variables that could skew results. This has led to a deeper understanding of how cells function in both health and disease, paving the way for groundbreaking discoveries. Their impact extends far beyond the lab, influencing clinical practices and patient care. Monoclonal cell lines are critical for the production of many life-saving drugs and therapies, ensuring that patients receive effective and safe treatments. Crafting clones, how monoclonal cell lines are born. The journey of creating these cell lines is both intricate and fascinating. Creating monoclonal cell lines is a multi-step process that requires precision and expertise. It begins with isolating a single cell with the desired characteristics. This step is crucial as it determines the quality of the final cell line. This cell is then stimulated to divide and multiply, a process that can take several days to weeks. This forms a colony of genetically identical cells, known as the monoclonal cell line. These colonies are essential for consistent and reliable research results. Several methods exist for isolating single cells, each with its own advantages and challenges. Limiting dilution is a common technique. It is straightforward, but requires careful handling to ensure accuracy. It involves diluting a cell suspension to deposit single cells into separate wells, allowing them to grow independently. Another method is fluorescence-activated cell sorting, or FACS. This technique is more advanced and allows for high precision. FACS uses fluorescent markers to identify and separate cells with specific properties, making it highly effective for isolating desired cells. Cloning rings physically isolate single cell colonies from a mixed population. This method is simple yet effective for certain applications. Each of these methods plays a vital role in the creation of monoclonal cell lines, contributing to advancements in medical research and biotechnology. A winning formula. Genetic stability and homogeneity. Two key characteristics make monoclonal cell lines so powerful. Genetic stability and homogeneity. Genetic stability means the cells retain their genetic makeup over time. This ensures consistent results in experiments and production processes. Homogeneity refers to the uniformity of the cells within the cell line. Since all cells originate from a single parent cell, they share the same characteristics and behavior. This uniformity is crucial for obtaining reliable and reproducible data in research and manufacturing. Medical marvels, monoclonal antibodies, and beyond. Monoclonal cell lines have revolutionized medicine, particularly in the field of immunotherapy. They are used to produce monoclonal antibodies, a type of targeted therapy used to treat a wide range of diseases, including cancer and autoimmune disorders. Monoclonal antibodies work by binding to specific proteins on the surface of cells. This binding can either block the activity of the protein or mark the cell for destruction by the immune system. The development of monoclonal antibody therapies has significantly improved outcomes for patients with previously untreatable diseases. Section 5. Protein powerhouses. Therapeutic proteins from cell factories. Beyond monoclonal antibodies, monoclonal cell lines serve as miniature factories for producing a variety of therapeutic proteins. These proteins, such as hormones, enzymes, and growth factors, are essential for treating various medical conditions. Insulin, used to treat diabetes, is a prime example. Monoclonal cell lines engineered to produce human insulin have revolutionized diabetes care. These cell lines provide a reliable and cost-effective method for producing large quantities of this life-saving hormone. Section 6, vaccines. Training the immune system with monoclonal cell lines. Monoclonal cell lines play a crucial role in vaccine development. These cell lines are derived from a single cell, ensuring uniformity and consistency in the production process. They are used to produce specific viral or bacterial proteins that can trigger an immune response without causing the disease. This method allows scientists to create vaccines that are both safe and effective. These proteins are then purified and formulated into vaccines. The purification process ensures that the final product is free from contaminants and safe for human use. By exposing the immune system to these harmless protein fragments, vaccines train the body to recognize and fight off the actual pathogen in the future. This training is crucial for building immunity and preventing outbreaks. This approach has been instrumental in eradicating diseases like smallpox and significantly reducing the incidence of others like polio and measles. The success of these vaccines highlights the importance of continued research and development in the field of immunology. Section 7. The genetic drift dilemma. Challenges in monoclonal cell lines. Despite their advantages, monoclonal cell lines are not without their challenges. One major concern is genetic drift. Over time, cells in culture can accumulate spontaneous mutations, leading to changes in their characteristics and behavior. Genetic drift can compromise the stability and homogeneity of a cell line. This can affect the reproducibility of experiments and the quality of therapeutic products derived from the cell line. Regular monitoring and careful maintenance are crucial to minimize the impact of genetic drift. Section 8. Time is of the essence. Overcoming the hurdles of development. Another challenge is the time and resources required to develop and characterize a new monoclonal cell line. The process of isolating a single cell, expanding it into a stable cell line, and validating its characteristics can take several months to a year. This lengthy development timeline can be a bottleneck in research and drug discovery. Researchers are constantly seeking ways to expedite the process without compromising the quality and stability of the resulting cell lines. Section 9. The future is automated. High-Throughput Technologies. Emerging technologies are addressing the challenges associated with monoclonal cell line development. High-throughput technologies, for instance, are revolutionizing the field by automating and accelerating many of the steps involved. These technologies allow researchers to screen thousands of cells simultaneously, significantly increasing the speed of identifying and isolating cells with desired properties. This speeds up the development process and enables the discovery of rare and valuable cell lines. Section 10. Microfluidics, a new wave in monoclonal cell line development. Microfluidics, the science of manipulating fluids at the microscale, is another promising area. Microfluidic devices allow researchers to culture and analyze cells in tiny, precisely controlled environments. This level of control enhances cell growth and allows for more sophisticated experiments. Microfluidics holds great potential for improving the efficiency and speed of monoclonal cell line development. Section 11. Looking ahead. The expanding potential of monoclonal cell lines. As technology advances, the potential applications of monoclonal cell lines continue to expand. Researchers are exploring their use in areas like personalized medicine, regenerative medicine, and disease modeling. In personalized medicine, monoclonal cell lines derived from a patient's own cells could be used to develop tailored therapies. In regenerative medicine, they hold promise for growing tissues and organs for transplantation. Section 12. A powerful tool. Monoclonal cell lines shape the future of medicine. Monoclonal cell lines are powerful tools that have revolutionized research and medicine. Their ability to produce specific proteins in cells has led to the development of life-saving therapies and vaccines. While challenges remain, emerging technologies are addressing these limitations and unlocking new possibilities. As our understanding of cell biology deepens and technology continues to evolve, monoclonal cell lines will undoubtedly play an even more significant role in shaping the future of medicine. Pause 3s. Section 13. Development of monoclonal cell lines. Available technologies and overcoming challenges. Single-cell cloning is the process by which a cell line is generated from a single starting cell, that has been segregated from a heterogeneous population, typically one that has been prepared from transfections, transductions, or primary cells from tissue or biopsy. As new methodologies evolve, life science drug development has begun to expand from small molecules toward more specific biologics and advanced therapy medicinal products, or ATMPs. Single-cell workflows have played an important role in this expansion, as they have clinical and scientific impacts on crucial cross-disciplinary applications in multi-omics, immune oncology, rare cell identification, drug discovery, stem cells, regenerative medicine, biologics, organoids, and epigenetics. In recent years, cell line development for the purpose of analytics, bioproduction, and therapeutics has evolved into a crucial workflow, with monoclonality as a critical requirement. The development of more specific therapies for a diverse range of disease conditions has also driven the advancement in single-cell workflow, particularly cloning, which has become a vital requirement in biopharmaceutical research and product development. While the journey is filled with challenges, the continuous evolution of technology and methodologies promises a brighter future for monoclonal cell lines, paving the way for innovative therapies and groundbreaking medical advancements. Innovations in single-cell isolation techniques. As we traverse the landscape of monoclonal cell line development, it is evident that traditional methods like limiting dilution and single-cell dispensing, while foundational, have their limitations. But the horizon is brightened by groundbreaking innovations designed to tackle these challenges head-on. Emerging technologies such as microfluidics and automated single-cell sorting are revolutionizing the field. Microfluidic devices allow for precise manipulation of individual cells in tiny volumes, vastly improving the efficiency and viability of isolated cells. These devices can sort and dispense single cells with remarkable accuracy, reducing the tediousness and potential errors inherent in manual techniques. Similarly, advances in automated single-cell sorting use sophisticated algorithms and high-throughput systems to identify and isolate viable monoclonal cells. These systems integrate powerful imaging technologies with robotics, ensuring that only the healthiest cells are selected for expansion. This not only accelerates the cloning process, but also enhances the reliability and reproducibility of the results. Another promising innovation is the use of CRISPR-Cas9 technology for precise genome editing within single cells. This allows for the creation of highly specific monoclonal cell lines tailored to particular research and therapeutic needs. The ability to edit genes at the single-cell level opens up new possibilities for developing targeted treatments and understanding complex biological processes. These advancements are not just incremental improvements. They represent a paradigm shift in how we approach single-cell isolation and monoclonal cell line development. By embracing these cutting-edge technologies, researchers and biopharmaceutical companies can overcome the traditional hurdles, paving the way for more efficient and reliable production of monoclonal cell lines. As we continue to innovate, the potential for monoclonal cell lines to transform medicine becomes ever more tangible. The future is not just about overcoming the challenges of today, but about pioneering the breakthroughs of tomorrow. Flow cytometry and cell sorting represent another set of techniques that promise improvements over traditional limiting dilution methods. These approaches analyze and sort cells based on hydrodynamic focusing and specific cellular characteristics. Fluorescence-activated cell sorting, or FACS, enables the isolation of single cells from a suspension with a certain degree of purity. Modern benchtop cell sorting systems, equipped with multiple lasers and colors, enhance the precision and speed of the sorting and selection process. However, this process requires significant gating requirements and setup for each population to be sorted, ensuring the retrieval of the maximum number of viable clones. Despite its advantages, FACS is not without challenges. The availability of fluorescently tagged antibodies for surface epitopes often complicates reagent preparation. Moreover, the sorting and dispensing process can physically stress the cells, altering their metabolic state and inducing oxidative stress. Studies indicate that only about 20% of isolated single cells from FACS produce usable colonies due to the fraction of sorted cells that are dead or have impaired growth. Additionally, FACS sorted cells exhibit a 50% increase in reactive oxygen species, highlighting the stress imposed during the transition from cell culture to the sorting environment. While flow cytometry and cell sorting offer advanced capabilities, they also underscore the need for continuous innovation to mitigate these challenges. The quest for more efficient and reliable single-cell isolation techniques remains a critical focus for researchers and biopharmaceutical companies alike. As we continue to push the boundaries of monoclonal cell line development, the integration of artificial intelligence, AI, and machine learning, ML, holds unprecedented promise. These advanced technologies are poised to revolutionize the way we approach single-cell isolation and clone generation. AI algorithms can analyze vast datasets generated from cell sorting processes, identifying patterns and optimizing parameters that human operators might overlook. Machine learning models, trained on historical data, can predict the best conditions for cell viability and growth, reducing the trial and error approach currently prevalent in the field. Moreover, AI-driven automation can streamline the gating and sorting setup in flow cytometry, minimizing human error and enhancing reproducibility. This can significantly reduce the physical stress on cells during sorting, potentially lowering the oxidative stress and increasing the survival rate of isolated single cells. By harnessing the power of AI and ML, researchers can achieve higher efficiency, greater accuracy, and more reliable outcomes in the development of monoclonal cell lines. This not only accelerates the pace of scientific discovery, but also brings us closer to innovative therapies and treatments for a myriad of diseases. The future of monoclonal cell line development is bright, and with AI and ML as our allies, we are poised to overcome the challenges and unlock new potentials in biopharmaceutical research and beyond. Stay tuned as we explore these cutting-edge advancements and their impact on the future of medicine. As we delve deeper into the realm of monoclonal cell line development, the landscape of cell sorting and dispensing technologies continues to evolve. Recently, several new products specializing in cell sorting and dispensing have emerged, each marketing their system as a method for streamlining the generation of single-cell clones. These setups vary widely in specifications and cost, ranging from $150,000 to millions of dollars. The most common of these systems are cell dispensers. These platforms use microfluidics combined with bright field imaging or fluorescence detection to deposit a single cell in one well of a 96 or 384 well collection plate. These platforms seek to improve upon limiting dilution techniques by eliminating the time and resources spent serially processing culture plates by directly obtaining an isolated single cell. This may provide some improvements over limiting dilution and improved confidence in monoclonality. However, such systems have limited benefits. Outgrowth of the isolated single cells must be performed separately, necessitating additional equipment, resources, and space demands. This leads to additional costs beyond the platform itself, which alone can run from several hundreds of thousands to millions of dollars in purchase price. The process of physical manipulation in fluidic channels and droplet impact of dispensing can also harm the isolated cell, perturbing expression profiles and reducing outgrowth. Furthermore, these platforms are specific in function and do not support other forms of selection or propagation. Researchers looking to propagate stem cells, iPSCs, organoids, or screen T cells cannot make use of them. Thus, although single-cell dispensers represent an improvement over traditional manual methods, they do not meet all needs to facilitate rapid and efficient development of clones. With the market saturated with a variety of platforms, researchers are left to navigate and decide which system is most appropriate and cost-effective for their needs. While new techniques such as microfluidic platforms and automated clone pickers show promise for the screening, selection, and isolation of monoclones, they still fall short on delivering a high volume of viable monoclonal colonies without constraining resources. There is a clear need for more comprehensive tools that encompass the entire workflow from single-cell separation through outgrowth, and that are applicable for multiple cell lines and types. Stay tuned as we continue to explore these advancements and seek out the next breakthrough in monoclonal cell line development. As we continue to navigate the complex landscape of cell sorting technologies, a clear need arises for systems that provide natural conditions to maintain cell cycle kinetics and ensure biochemical equilibrium during isolation. Introducing the CellRaft technology, a breakthrough designed to meet these unmet needs. The CellRaft AIR system is an integrated platform for growing, scanning, analyzing, and isolating single-cell-derived monoclonal colonies. Utilizing the CellRaft AIR array, a cell culture dish with 10,000 to 150,000 microwells called CellRafts, this technology allows cells to settle gently by gravity, distributing into single, double, or clustered combinations. In this system, approximately 40% to 60% of cell rafts are populated by single cells, depending on seeding density. The design ensures all cells share the same media and extracellular growth factors or cytokines, mimicking the conditions of an actual flask or reactor. This eliminates physical perturbation, maintaining biochemical and physiological stability. The CellRaft AIR system enables imaging and analysis of cells in bright field or fluorescent modalities. With the CellRaft Cytometry (TM) software, users can select clones that precisely fit defined attributes. The system automates acquisition, isolation, and retrieval of monoclonal colonies using a magnetic wand, gently transferring colony-containing cell rafts to a 96-well collection plate. This versatile system supports colony growth from primary, adherent, or suspension cells, including iPSCs and immune cells. It also enables the growth of iPSCs into 3D cell systems, such as organoids, for advanced applications in cancer immunology, multi-omics, and cellular heterogeneity studies. By providing flask-like conditions at the resolution of a single cell, the CellRaft AIR system ensures the viability and vitality of single cells, allowing them to develop into healthy clones. This represents a significant improvement over traditional systems, promoting the selection of cells that are not in a resting phase, early apoptosis, or senescence. The CellRaft AIR system embodies a new wave in monoclonal cell line development, offering comprehensive tools that encompass the entire workflow from single-cell separation through outgrowth. This innovation paves the way for obtaining 10x to 50x more viable, highly proliferative monoclonal colonies, shaping the future of medicine. Stay tuned as we delve deeper into these advancements, continually seeking the next breakthrough in monoclonal cell line development. As we look to the future, the advancements in monoclonal cell line development hold the promise of revolutionizing personalized medicine. With the emergence of technologies like the CellRaft AIR system, we are on the cusp of achieving unprecedented precision in the selection and cultivation of monoclonal cell lines. These innovations not only enhance the viability and proliferation of cells, but also open new frontiers in therapeutic applications. Imagine a world where treatments are tailored to the genetic and cellular makeup of each individual patient. Monoclonal cell lines serve as the foundation for this personalized approach, enabling the creation of therapies that are specifically designed to target the unique characteristics of a person's disease. Whether it's cancer immunotherapy, regenerative medicine, or advanced diagnostics, the potential applications are limitless. The CellRaft AIR system, with its ability to maintain natural cell conditions and automate the selection process, ensures that only the healthiest and most viable cells are chosen for further development. This level of precision reduces the risk of treatment failure and increases the likelihood of positive outcomes for patients. Furthermore, the integration of artificial intelligence and machine learning with cell sorting technologies promises to accelerate the discovery of new treatments. By analyzing vast amounts of cellular data, these advanced algorithms can identify patterns and insights that were previously beyond our reach. This data-driven approach will not only speed up the development of new therapies, but also improve their efficacy and safety. As we continue to push the boundaries of what's possible, the collaboration between scientists, clinicians, and technologists will be crucial. Together, we can harness the power of monoclonal cell lines to transform the landscape of medicine, bringing hope and healing to millions of people around the world. Join us in the next chapter as we explore the cutting-edge research and groundbreaking discoveries that are shaping the future of personalized medicine. The journey has just begun, and the possibilities are endless. Stay tuned. Similar to standard culturing methods, cells are plated on the CellRaft array, which is then loaded into the CellRaft AIR system platform. Cells are imaged with 3-channel fluorescence and bright field microscopy and sorted with user-defined thresholds, filtering, and gating. The software will scan for expression, time, morphology, and automatically isolate the desired cell raft based on user criteria. The system can isolate a full 96-well plate of individual, undisturbed cells or colonies in under an hour for expansion and downstream analysis. The CellRaft AIR system offers a number of advantages over other currently available systems. It provides a more efficient single-cell workflow, including clonal colony propagation, by combining imaging, identifying, and isolating in one instrument and on one consumable. It achieves this while also producing very high clonal yields with robust viability and cell proliferation. This is a significant improvement over other platforms, which often have additional space and instrument needs. Furthermore, automation of all steps provides significant benefits, including real-time imaging, identification, and isolation of cells and small colonies for outgrowth in 96 well plates. This not only accelerates the timeline to results, but also reduces costs and contamination risks. The system includes several onboard assays, including cell characterization, co-culturing, cell drug, and cell-cell interactions. Finally, unlike other systems, the CellRaft AIR system supports a wide range of cell types and 3D cell systems, including stem cells, including iPSCs, animal or human cells, primary cells, immortalized cells, adherent cells, suspension cells, and organoids. Recent experimental and cost analyses, including hands-on time to run the protocol between limiting dilution and CellRaft technology, indicate that the latter delivers a high return on investment in terms of outgrowth efficiency, time, and cost. Currently, single-cell workflows for approximately 100 different cell lines and types have been successfully demonstrated on the CellRaft AIR system, and new workflows are being constantly developed and validated. As we embrace these cutting-edge technologies, the horizon of personalized medicine expands even further, bringing the promise of tailored therapies closer to reality. Join us as we continue to explore the forefront of medical advancements in our next chapter. For decades, limiting dilution and single-cell sorting have been the primary methods for development of monoclonal cell lines. Significant disadvantages of cellular damage, slow workflows, and questions of clonality have remained outstanding problems with these methods. Newer single-cell dispensers have provided significant improvements in assurance of clonality and confluence, but yields can still be low due to cellular perturbation from the selection process. Furthermore, there are new demands for a wider array of selection capabilities for stem cells/iPSCs, organoids, adherent cells, and rare cell types. The CellRaft AIR system is an integrated platform that encompasses imaging, tracking, analysis, and automated isolation of verified monoclonal cultures. It offers significant cost saving, viability, and high outgrowth advantages over other systems that require additional space, equipment, and manual labor. The CellRaft AIR system is robust for numerous applications such as cell line development, CRISPR gene editing, stem cell culture, organoid development, and single-cell genomics, making it ideal for a wide range of fields. Ultimately, this platform may bring unparalleled benefits to the field by streamlining and speeding the drug development process, helping to bring drugs to market more rapidly. Imagine a world where personalized medicine is not a distant future, but a present reality. The CellRaft AIR system is not just a tool. It's a transformative technology that is paving the way for the next generation of medical breakthroughs. By addressing the limitations of traditional methods and meeting the demands of modern research, it stands as a beacon of innovation in the landscape of monoclonal cell line development. As we continue to embrace these advancements, the horizon of personalized medicine expands even further, bringing the promise of tailored therapies closer to reality. Stay with us as we delve deeper into the cutting-edge technologies shaping the future of medicine. Monoclonal cell lines play a critical role in advancing stem cell research by providing a controlled, uniform, and genetically stable population of cells for various applications. The development of monoclonal cell lines from stem cells allows researchers to investigate fundamental biological processes, such as cellular differentiation, tissue regeneration, and disease modeling. Stem cell research holds great promise for regenerative medicine, therapeutic development, and understanding developmental biology, and monoclonal cell lines contribute significantly to these efforts. In this comprehensive discussion, we will explore the role of monoclonal cell lines in stem cell research through the following sections. 1. Introduction to stem cells and monoclonal cell lines, definitions and key concepts, importance of monoclonality in stem cell research. 1. Methods of generating monoclonal stem cell lines, techniques for isolation of single stem cells, expansion and cloning of stem cells. 1. Applications of monoclonal stem cell lines in research, studying cellular differentiation, disease modeling and drug screening, regenerative medicine and tissue engineering, gene editing and functional genomics. 1. Monoclonal cell lines from different types of stem cells, embryonic stem cells, ESCs, induced pluripotent stem cells, iPSCs, Adult stem cells, ASCs. 1. Challenges and limitations in developing monoclonal stem cell lines. Genetic and epigenetic stability. Technical challenges in cloning and culture. 1. Emerging technologies in monoclonal stem cell line research. CRISPR/Cas9 and gene editing tools. Organoid development and 3D culture systems. 7. Ethical considerations. Ethical issues in stem cell research. The role of monoclonal cell lines in ethical research. As we continue to push the boundaries of stem cell research, monoclonal cell lines will remain a cornerstone of innovation, offering unparalleled opportunities to unlock the mysteries of human biology and develop groundbreaking therapies. Join us as we delve deeper into this fascinating field, exploring the cutting-edge technologies and ethical considerations shaping the future of medicine. As we delve deeper into the world of monoclonal stem cell research, it's essential to address the ethical considerations that accompany such groundbreaking advancements. The development and application of monoclonal cell lines from stem cells bring forth a myriad of ethical questions that must be navigated with care and responsibility. The primary ethical concerns revolve around the sources of stem cells, particularly embryonic stem cells, ESCs. The use of ESCs involves the destruction of human embryos, which raises significant moral and philosophical debates. Some argue that the potential benefits, such as curing debilitating diseases and understanding human development, justify the use of ESCs. Others believe that alternative sources, like induced pluripotent stem cells, iPSCs, and adult stem cells, ASCs, should be prioritized to avoid ethical dilemmas. Induced pluripotent stem cells, iPSCs, offer a promising solution by reprogramming adult cells to a pluripotent state without the use of embryos. This technology not only addresses ethical concerns, but also provides a patient-specific approach to stem cell therapy, minimizing immune rejection risks. However, the long-term safety and efficacy of iPSCs remain under scrutiny. Adult stem cells, ASCs, present another ethically favorable option, as they can be obtained from the patient's own tissues, such as bone marrow or adipose tissue. While ASCs have more limited differentiation potential compared to ESCs and iPSCs, their use circumvents the ethical issues associated with embryonic stem cells. Moreover, the ethical landscape extends to the potential applications of monoclonal stem cell lines. The prospect of gene editing, using tools like CRISPR-Cas9, raises questions about the implications of genetic modifications in humans. The possibility of creating designer babies or enhancing human traits through genetic engineering sparks debates regarding the boundaries of scientific intervention. In addition, the development of organoids and 3D culture systems from monoclonal stem cell lines opens new avenues for disease modeling and personalized medicine. However, these advancements also pose ethical challenges related to the creation of complex, organ-like structures that may possess some level of consciousness or sensory perception. As we navigate these ethical landscapes, it is crucial to establish robust regulatory frameworks and ethical guidelines that balance scientific progress with moral responsibility. Engaging in transparent, inclusive discussions with stakeholders, including scientists, ethicists, policymakers, and the public, will ensure that the advancements in monoclonal stem cell research are guided by ethical principles and societal values. Join us in the next section as we explore the emerging technologies that are shaping the future of monoclonal stem cell research and their potential to revolutionize medicine. As we delve into the cutting-edge techniques employed in generating monoclonal stem cell lines, it's essential to understand the meticulous processes that underpin this remarkable field of research. The journey begins with the isolation of a single stem cell from a diverse population. Let's explore the primary methods used to achieve this. Firstly, we have limiting dilution. This technique involves plating stem cells at exceptionally low densities to isolate individual cells in separate wells. It's a method frequently utilized for creating monoclonal lines from pluripotent stem cells. Next is fluorescence-activated cell sorting, or FACS. FACS employs fluorescent markers to identify and sort individual stem cells based on their surface markers. This technique is particularly valuable when specific subpopulations of stem cells need to be isolated with precision. Another key technique is magnetic activated cell sorting, or MAX. MAX uses magnetic beads conjugated to antibodies that bind to specific cell surface markers. This method allows for the isolation of stem cells from a mixed population based on their distinct markers. Once a single stem cell is successfully isolated, the next phase involves its expansion and cloning. Culturing the isolated stem cell under controlled conditions is crucial to support its growth into a monoclonal population. This process requires a delicate balance between promoting cell proliferation and maintaining the stem cell's pluripotency or multipotency. Researchers must carefully select the appropriate growth factors and culture conditions to ensure that the cells remain undifferentiated and capable of self-renewal. However, achieving high cloning efficiency can be challenging. Stem cells, particularly pluripotent stem cells, are sensitive to environmental conditions, and not all isolated single cells will successfully form colonies. Fortunately, advancements in stem cell culture techniques, such as the use of feeder layers and optimized growth media, have significantly improved cloning success rates. These innovations are paving the way for more efficient and reliable generation of monoclonal stem cell lines. Join us in the next section as we uncover the emerging technologies that are revolutionizing the field of monoclonal stem cell research and their potential to transform the future of medicine. Monoclonal stem cell lines provide a controlled and reproducible system for a wide range of research applications. Here, we will explore some of the most important uses of these cell lines. One of the primary roles of monoclonal stem cell lines in research is to study the mechanisms of cellular differentiation. By isolating and expanding monoclonal lines, researchers can track how individual stem cells differentiate into specific cell types, such as neurons, cardiomyocytes, or hematopoietic cells. This is particularly important for understanding the signaling pathways, transcription factors, and gene networks that govern cell fate decisions. For example, monoclonal iPSC lines can be differentiated into specific cell types to model developmental processes in vitro. By using monoclonal lines, researchers can ensure that the differentiation process is consistent across multiple experiments, allowing for a more precise understanding of how stem cells give rise to various tissues. Monoclonal stem cell lines are invaluable for creating disease models in vitro. By using gene editing techniques such as CRISPR-Cas9, researchers can introduce disease-associated mutations into monoclonal stem cell lines. These cells can then be differentiated into relevant cell types, such as neurons for neurodegenerative disease models or cardiomyocytes for heart disease models. This enables the study of disease mechanisms in a controlled and reproducible system. Moreover, monoclonal stem cell lines can be used for high-throughput drug screening. In this context, researchers can test potential therapeutic compounds on differentiated cells derived from monoclonal stem cell lines to evaluate their efficacy and toxicity. This approach is particularly useful for personalized medicine, where patient-specific iPSCs can be generated and drug responses can be tested on a genetically matched cellular model. In regenerative medicine, monoclonal stem cell lines offer a reliable source of cells for potential therapeutic applications. For example, in the field of tissue engineering, monoclonal stem cell lines can be differentiated into specific cell types, such as skin cells, cartilage cells, or bone cells, which can then be used to create functional tissues or organs. Monoclonal lines derived from iPSCs are particularly promising for autologous cell therapies, where a patient's own cells are reprogrammed into iPSCs, expanded as a monoclonal line, and differentiated into the desired cell type for transplantation. This approach minimizes the risk of immune rejection and has shown great potential for treating conditions such as spinal cord injuries, heart disease, and diabetes. Monoclonal stem cell lines are also used for gene editing and functional genomic studies. By using gene editing tools like CRISPR-Cas9, researchers can introduce specific genetic modifications into monoclonal stem cell lines to study gene function, regulatory elements, and the effects of mutations. These modified monoclonal lines can then be differentiated into various cell types to investigate how the genetic changes impact cellular behavior and development. This approach has been especially useful in identifying the genetic basis of human diseases, as well as in validating drug targets. For example, researchers can generate monoclonal stem cell lines with knockouts of specific genes and study how these genes affect cell differentiation and function. Through these diverse applications, monoclonal stem cell lines are driving significant advancements in biomedical research, offering new insights into cellular processes, disease mechanisms, and potential therapies. Stay tuned as we continue to explore the vast potential of monoclonal cell lines in shaping the future of medicine. As we look to the future, the potential of monoclonal stem cell lines in medicine is boundless. These lines are not just tools for understanding biology, they are the building blocks of tomorrow's therapies and medical innovations. With the advent of advanced gene editing techniques like CRISPR/Cas9 and the integration of artificial intelligence, the development of monoclonal stem cell lines is poised to revolutionize personalized medicine. Imagine a world where a patient's own cells can be reprogrammed and tailored to treat their specific condition, offering precise and effective treatments with minimal side effects. In the realm of regenerative medicine, monoclonal stem cell lines hold the promise of regenerating damaged tissues and organs. Researchers are already making strides in creating functional tissues, such as heart muscle cells for repairing heart damage, or neural cells for treating neurodegenerative diseases. The ability to grow these tissues from a patient's own cells means that we can potentially eliminate the risk of immune rejection, a major hurdle in transplantation medicine. Moreover, the use of monoclonal stem cell lines in drug discovery is accelerating the development of new treatments. By creating disease-specific cell models, researchers can screen thousands of compounds to find potential drugs more efficiently. This not only speeds up the process of finding effective treatments, but also reduces the reliance on animal models, making the research more ethical and directly relevant to human health. However, the journey is not without its challenges. Ensuring the genetic and epigenetic stability of these cells, overcoming technical difficulties in reprogramming and differentiation, and addressing the ethical considerations are all critical steps that need to be navigated carefully. But with continued innovation and collaboration across scientific disciplines, the future of monoclonal stem cell lines looks incredibly promising. As we stand on the brink of these medical breakthroughs, one thing is clear: monoclonal stem cell lines will play a pivotal role in shaping the future of medicine, offering hope and healing to countless individuals around the world. Stay with us as we explore how these advancements are already beginning to transform the landscape of healthcare, bringing us closer to a future where personalized, regenerative, and highly effective treatments are the norm. As we delve further into the future of monoclonal stem cell lines, it's essential to highlight the emerging technologies that are transforming this field. These advancements are enabling more precise manipulation and investigation of stem cell behavior, pushing the boundaries of what we thought possible. CRISPR/Cas9 and gene editing tools. One of the most revolutionary technologies in recent times is CRISPR-Cas9. This gene editing tool allows for precise modification of the genome at specific locations. In the context of monoclonal stem cell line research, CRISPR-Cas9 offers the ability to introduce targeted mutations, correct genetic defects, or insert reporter genes into monoclonal lines. This capability is invaluable for creating disease models, studying gene function, and exploring the role of specific genetic variants in cellular differentiation. By combining CRISPR-Cas9 with monoclonal stem cell lines, researchers can create isogenic cell lines. These lines differ by only a single genetic change, enabling more precise studies of how specific genes influence stem cell behavior, disease progression, or drug response. Organoid development and 3D culture systems. Another groundbreaking advancement is the development of organoids, three-dimensional structures derived from stem cells that mimic the architecture and function of organs. Organoids represent a significant leap in tissue engineering and regenerative medicine, offering more physiologically relevant models than traditional 2D cell cultures. Monoclonal stem cell lines are crucial for organoid development, providing a uniform starting population of cells that can be directed to form specific tissues. This consistency allows researchers to generate organoids that are genetically and phenotypically uniform, facilitating more accurate studies of tissue development, disease mechanisms, and drug responses. Moreover, advances in 3D culture systems, such as bioprinting and microfluidic platforms, are further enhancing the ability to create complex tissue models from monoclonal stem cell lines. These technologies hold immense promise for applications in personalized medicine, where patient-specific monoclonal lines can be used to generate functional tissue constructs for disease modeling or therapeutic purposes. These emerging technologies are not just tools, they are the catalysts driving the next wave of innovation in monoclonal stem cell line research. As we harness these advancements, the potential for groundbreaking discoveries and transformative therapies becomes increasingly within reach. Stay with us as we continue to explore the challenges and limitations in developing monoclonal stem cell lines, and how these cutting-edge technologies are helping to overcome them. Monoclonal cell lines play a pivotal role in organoid research, providing a homogeneous and genetically consistent source of cells that can differentiate into complex, three-dimensional structures mimicking human organs. Organoids, which are miniature, self-organizing structures derived from stem cells, have become valuable models for studying human development, disease mechanisms, and drug responses. The use of monoclonal stem cell lines ensures that these organoids are genetically uniform, reproducible, and reliable for scientific investigations. To understand the role of monoclonal cell lines in organoid research, it's important to examine the following key areas. 1. Consistency and reproducibility in organoid formation. Monoclonal cell lines provide a uniform starting point, ensuring that the resulting organoids are consistent in their genetic makeup and structural characteristics. This consistency is crucial for generating reliable data and reproducible results in research. 1. Modeling human development and organogenesis. By using monoclonal stem cell lines, researchers can create organoids that closely mimic the developmental processes and organ formation seen in humans. This allows for detailed studies of early human development and the identification of key factors involved in organogenesis. 1. Disease modeling and personalized medicine. Monoclonal cell lines enable the creation of disease-specific organoids, allowing scientists to study the progression and underlying mechanisms of various diseases. Additionally, patient-derived monoclonal cell lines can be used to generate personalized organoids, which can be employed to test potential treatments and develop tailored therapeutic strategies. 1. Drug screening and toxicity testing. Organoids derived from monoclonal cell lines provide a more accurate and physiologically relevant model for drug screening and toxicity testing compared to traditional cell cultures. This enhances the predictive power of preclinical studies and supports the development of safer and more effective drugs. 1. Challenges and future directions. Despite the significant advancements, there are still challenges to overcome in organoid research. These include improving the scalability of organoid production, enhancing the complexity and functionality of organoids, and integrating organoid models into clinical applications. Continued research and technological innovations are essential to address these challenges and unlock the full potential of monoclonal cell lines in organoid research. As we continue to explore these fascinating areas, the impact of monoclonal cell lines on organoid research becomes increasingly evident, paving the way for groundbreaking discoveries and transformative medical advancements. Stay with us as we delve into the ethical considerations in monoclonal stem cell research, and how these cutting-edge technologies are helping to overcome the challenges. As we continue to push the boundaries of science, it's imperative to address the ethical considerations intertwined with monoclonal stem cell research. This scene will delve into the ethical landscape, exploring both the challenges and the solutions that monoclonal cell lines offer. Ethical issues in stem cell research. Stem cell research, particularly when involving human embryonic stem cells, raises significant ethical questions. The most prominent issue surrounds the use of human embryonic stem cells, ESCs, which are derived from human embryos. The destruction of embryos to obtain ESCs raises concerns for many, particularly from religious and philosophical perspectives. In response, various countries have implemented strict regulations regarding the derivation and use of ESCs. Moreover, the use of genome editing in stem cells, especially in human germline editing, poses ethical dilemmas regarding the potential for unintended consequences, the transmission of genetic modifications to future generations, and the possibility of designer babies. These concerns necessitate a careful and measured approach to ensure responsible scientific advancement. The role of monoclonal cell lines in ethical research. The development of induced pluripotent stem cells, iPSCs, from adult somatic cells has alleviated many of the ethical concerns associated with embryonic stem cells. iPSCs offer a source of pluripotent stem cells without the need for embryos, making them more ethically acceptable. Monoclonal iPSC lines allow researchers to conduct stem cell research and develop regenerative therapies without the ethical issues tied to the destruction of human embryos. Furthermore, monoclonal cell lines provide a consistent and reproducible model for studying diseases, testing drugs, and developing therapies. This uniformity reduces the variability and uncertainty often associated with stem cell research, leading to more reliable and ethically sound scientific outcomes. As we navigate the complex ethical landscape of stem cell research, monoclonal cell lines present a promising path forward. They not only advance our scientific understanding, but also uphold ethical standards, paving the way for responsible and impactful innovations in medicine. Stay with us as we explore the challenges and limitations in developing monoclonal stem cell lines and how these cutting-edge technologies are helping to overcome them. Despite their many advantages, developing monoclonal stem cell lines comes with a range of challenges, particularly concerning their genetic stability, epigenetic integrity, and technical difficulties in isolating and expanding single cells. Maintaining genetic and epigenetic stability in monoclonal stem cell lines is crucial, as even minor changes can lead to alterations in cellular behavior, differentiation potential, or disease modeling outcomes. During prolonged culture, stem cells are susceptible to genetic mutations and chromosomal abnormalities that can accumulate over time. This phenomenon, known as genetic drift, can result in inconsistent or unreliable results. Additionally, epigenetic modifications, such as DNA methylation and histone modifications, can occur during cell culture, affecting gene expression without altering the underlying DNA sequence. These changes can influence stem cell pluripotency or differentiation potential, making it difficult to obtain reproducible results. To mitigate these risks, researchers use various strategies to monitor and preserve the genetic and epigenetic integrity of monoclonal stem cell lines. Techniques such as karyotyping, whole genome sequencing, and epigenetic profiling are employed to ensure that the cells remain genetically stable over time. However, maintaining epigenetic stability remains a significant challenge, particularly for long-term culture of pluripotent stem cells. Isolating and expanding single stem cells to generate monoclonal lines is technically challenging. Stem cells, particularly pluripotent stem cells, are highly sensitive to their microenvironment, and small changes in culture conditions can affect their survival, proliferation, and differentiation potential. Creating an environment that supports the growth of a single cell while maintaining its stemness is difficult, and not all single cells will successfully form colonies. Additionally, stem cells often exhibit low cloning efficiency, meaning that a significant proportion of isolated single cells fail to proliferate and form monoclonal populations. To address this, researchers use specialized culture media, growth factors, and feeder cells that provide the necessary support for stem cell expansion. Nevertheless, achieving consistent and efficient cloning remains a key obstacle in stem cell research. Organoids derived from monoclonal stem cell lines offer a unique opportunity to model human development and organogenesis in vitro. These models allow researchers to observe how stem cells differentiate into specific cell types and organize themselves into complex structures resembling human organs. Monoclonal cell lines provide a controlled system to study the genetic and molecular mechanisms that regulate early developmental processes. For example, monoclonal embryonic stem cell, ESC, or induced pluripotent stem cell, iPSC, lines can be directed to differentiate into various organ-specific cell types, such as hepatocytes, liver cells, neurons, brain cells, or epithelial cells, intestinal cells. These cells can then self-organize into 3D organoids, recapitulating the structural and functional features of the corresponding human organ. Using monoclonal lines ensures that all cells within the organoid share the same genetic makeup, making it easier to dissect the pathways involved in differentiation and morphogenesis. This level of control is essential for understanding how specific genetic mutations or signaling pathways influence organ development, and can help identify critical factors involved in congenital diseases or developmental disorders. Monoclonal stem cell lines are invaluable for creating disease-specific organoid models, especially in the context of personalized medicine. By generating monoclonal iPSC lines from patients with genetic diseases, researchers can produce organoids that accurately reflect the patient's genetic background and disease pathology. These patient-specific organoids serve as powerful tools for studying the progression of diseases and testing potential therapies in a controlled, reproducible environment. For instance, iPSCs from a patient with cystic fibrosis, CF, can be used to generate lung organoids. Using monoclonal iPSC lines ensures that the organoids exhibit uniform disease characteristics, enabling researchers to study how CF affects lung tissue development and function. Furthermore, these organoids can be used to test personalized treatments, such as gene therapies or small molecule drugs, to evaluate their efficacy and safety in a patient-specific context. Monoclonal organoids also provide a valuable platform for studying complex polygenic diseases such as cancer. Cancer organoids derived from monoclonal cell lines can be used to model tumor growth, metastasis, and response to therapies. The genetic consistency of monoclonal cell lines ensures that researchers can track the effects of specific mutations or treatments on tumor progression in a highly controlled setting, allowing for more accurate predictions of therapeutic outcomes. In the realm of organoid research, the ability to generate organoids that consistently replicate the architecture and function of specific organs is paramount. This consistency is critical for both research and therapeutic applications. Monoclonal cell lines, derived from a single progenitor cell, provide a genetically stable and homogeneous population of cells. This uniformity is essential because it reduces variability in the organoid formation process, leading to more consistent results across experiments. In organoid research, variations in cell populations can lead to inconsistencies in organoid structure, function, and behavior. Monoclonal cell lines eliminate this issue by ensuring that all cells within the culture are genetically identical. This reproducibility is particularly important when studying complex processes such as tissue development or drug responses, as it allows for controlled comparisons between different experimental conditions. For instance, when generating brain organoids to study neurodevelopmental disorders, using monoclonal stem cell lines ensures that all organoids develop similarly. This uniform development allows researchers to pinpoint specific changes related to the disease or treatment being studied. In contrast, the use of heterogeneous cell populations would introduce variability that could obscure the effects of the experimental conditions. By harnessing the power of monoclonal cell lines, scientists can achieve a new level of precision and reliability in organoid research, paving the way for groundbreaking discoveries and innovative therapies. As we delve deeper into the realm of organoid research, the potential for personalized medicine becomes increasingly tangible. Monoclonal cell lines, with their genetic uniformity, serve as a cornerstone for creating organoids tailored to individual patients. This breakthrough allows for the development of personalized treatment plans that are both precise and effective. Imagine a patient diagnosed with a rare genetic disorder. Using their own cells, scientists can generate monoclonal organoids that accurately replicate the affected tissue. These personalized organoids can then be used to test various drugs and therapies, identifying the most effective treatment with minimal side effects. This process not only accelerates the path to recovery, but also significantly reduces the trial and error period typically associated with finding the right medication. Moreover, personalized organoids can be instrumental in understanding how a patient's unique genetic makeup influences their response to different treatments. For instance, cancer patients can benefit immensely from this approach. By generating tumor organoids from their own cancer cells, oncologists can test multiple chemotherapy agents to determine which one targets the tumor most effectively, thereby tailoring the treatment to the patient's specific needs. The advent of personalized medicine using monoclonal organoids marks a significant leap forward in healthcare. It promises a future where treatments are no longer one-size-fits-all, but are instead customized to the individual's genetic profile, leading to better outcomes and improved quality of life. As we stand on the brink of this medical revolution, the potential for monoclonal organoids in personalized medicine is boundless. With continued research and innovation, we are poised to transform the landscape of healthcare, making personalized treatment a reality for all. One of the most promising applications of organoid research is its use in drug discovery and toxicity testing. Organoids derived from monoclonal cell lines offer a physiologically relevant model for testing the effects of new drugs on human tissues. These 3D structures more closely mimic the cellular architecture and microenvironment of human organs than traditional 2D cell cultures, making them a superior platform for evaluating drug efficacy and safety. Monoclonal cell lines ensure that the organoids used in drug screening are genetically uniform, reducing variability and enabling researchers to obtain consistent results across multiple experiments. This uniformity is critical for identifying the specific effects of a drug on a particular tissue or organ. For example, Liver organoids derived from monoclonal iPSC lines can be used to screen drugs for hepatotoxicity, liver toxicity. Since all the cells in the organoid share the same genetic background, any observed toxicity can be directly attributed to the drug being tested, rather than to genetic variations within the cell population. Similarly, brain organoids derived from monoclonal lines can be used to evaluate the neurotoxicity of new compounds, providing a more accurate prediction of their potential side effects in humans. Monoclonal organoids also allow for high-throughput drug screening, where large libraries of compounds can be tested for therapeutic potential. By ensuring that each organoid is genetically identical, researchers can perform parallel experiments and compare the effects of different drugs in a reproducible manner. In conclusion, monoclonal cell lines have revolutionized the field of biotechnology and medicine. From their creation to their application in therapeutic proteins, stem cell research, and personalized medicine, these cell lines offer immense potential. As we continue to innovate and overcome challenges, the future of medicine looks promising. Thank you for joining us on this deep dive into the world of monoclonal cell lines. If you found this video informative, please like, share, and subscribe to our channel for more insightful content.